welcome back to online NPTEL course on Introduction to Complex Biological System. And currently we are discussing about infectious diseases, particularly virus and bacteria, and this is module 9. So today in this lecture I will be mostly discussing about different aspects of bacteria, and here particularly I will focus on different types of shapes, arrangement and sizes of bacteria, followed by the organization of bacterial cell and bacterial cell membrane and cell wall. Both are very important for them, and also some special features of bacteria. Now since this course name is Introduction to Complex Biological System, I would like to clarify here, although bacteria, they are just unicellular, they are prokaryotes, but still you will see a lot of complexities are present in bacteria, and these complexities are sometimes evolving also, and that is a little bit problem for us, because if you consider that pathogenic bacteria, for example, if they are evolving, and that is a problem for us to tackle them, right? So for example, many bacteria over the time they are becoming antibiotic resistance. This is just one example, but there are many examples like this, and that's why the field of microbiology, this is constantly evolving, and to take care of this issue we need to know like little about their structure, function, relation of macromolecules present in bacteria, and I'm going to discuss a few of them. Here, if you concentrate on shape, arrangement and size of bacteria, this is just to compare that where we are now, like compared to mammalian cell, compared to eukaryotic cell, something like that. So here in this diagram, I am trying to explain the size of bacteria relative to viruses. As you know, virus, they are just particles, some kind of virus particle we say, and now viruses are much smaller compared to bacteria. Now very common laboratory strain, for example E. coli, so its dimension mentioned here 1300 by 4000 nanometer. So this is, roughly this is the dimension of E. coli. This is E. coli. Okay. So as a result of that, I would say 1.3 micrometer, here 4 micrometer, and here whatever dimension we have mentioned, either its diameter or width by length, because some cases if bacteria is not like long enough, it's kind of more or less spherical, then we are mentioning in terms of diameter. As you can see, Streptococcus, it's a spherical, almost spherical bacteria. So here the diameter is 800 to 1000 nanometer. And now if we compare that bacteria with some viruses, for example pox virus, it's roughly bigger in size compared to other viruses, as you can see here. And on the other hand, influenza virus is much smaller. It's more or less spherical in nature, as you can see. This is influenza virus and 85 nanometer in diameter. And one of the smallest virus is poliovirus. As you can see, like almost we cannot see here something is there, some like small dot here, and only 27 nanometer diameter, right? And similarly some other viruses are also mentioned here. This is just to get a scale how their size really in terms of comparison between virus and bacteria. Here I would like to mention for example if you consider one RBC, so this is red blood cell. I am drawing in this way because no nucleus there and its shape is a little bit different. So RBC, the diameter of RBC is about 7000 nanometer. So now you can compare this with E. coli, and although I should mention here RBC, they are roughly, I would say they are kind of smaller cells compared to other cells present in our body. Most of the other cells present in our body, their dimension should be in between, I would say, 10 micrometer to 100 micrometer, in this range. Most of the other cells present in our body, and this is RBC, red blood cell. And now, since we discussed about DNA also in the first module, so now if I draw a DNA here, so what is the width of DNA? This is just 2 nanometer. Now I believe that you have kind of overall idea about the dimension of virus, bacteria, eukaryotic cell, and for a scaling purpose you can consider the width of DNA, which is just 2 nanometer. Now these small bacteria, they organize in different ways. Different type of organizations are also available. That is one thing. And the other thing, although they are small, but still they have different types of shapes. So we are going to discuss in this slide about different types of shapes and organization of bacteria. Now here this is cocci, or in singular form it's called coccus. So there are many bacteria which are belong to this group cocci. Their shape is roughly spherical. I would say it is roughly spherical in shape. And if I give you one example, for example, like Staphylococcus. This is very common bacteria, and then Streptococcus. So these are the coccus bacteria. So in their name, in the genus, you can see at the end it's coccus there. So they are roughly spherical bacteria, but they can stay just like in single spherical object like this, or sometime they can form different types of higher order structure. For example, some coccus, they can be paired like in this way, like 2 coccus, they are together. During division, somehow they are staying together. So this is the example on bacteria I can say here, Diplococcus. So this is 2 coccus, they are staying together, Diplococcus. Not just 2, there are some other bacteria, they can be in some kind of chain. As you can see here, this is in chain. So although they are coccus, but they are in chain, and this is Streptococcus. They are in chain. And similarly, so this chain form, because when they are dividing in only one plane, and they are not getting separated. So as a result of that, although they are single cell, but they are forming some kind of chain like structure. Similarly, some other bacteria, for example, Staphylococcus, so they divide in multiple plane and they are not getting separated. So as a result of that, they are just staying as just like grapes. So together, like multiple cells together. So this is called grapes like structure, and Staphylococcus aureus, they can form this kind of arrangement. And now if we move to another form of bacteria, for example, bacilli, or in singular bacillus, and one very common example is E. coli, Escherichia coli, and another common example, like all of you know its name, Mycobacterium tuberculosis. And there are many species of bacillus. So in their name itself, the bacilli is there. So all of them, they are kind of slender like rod like structure, and this is called bacillus. And here you can see this is bacilli in chain. So again, during division, they are not getting separated, so they are staying in chain. So this is these two, like cocci and bacilli, they are the most common form of bacteria in terms of their shape. But there are some deviation from this standard format, like coccus and bacillus. As you can see, this is one bacteria. It's like looks like comma shaped. It's comma shaped, right? So comma shaped. Now this is one example is Vibrio cholerae. Vibrio cholerae is a bacteria, and this looks like this comma shaped bacteria. And apart from that, as you can see here, their shape are somewhat like spiral shaped bacteria. And this spiral shaped bacteria, they have some kind of advantage also for them, because of, as you can see, this is almost like a screw. So as a result of that, it helps their motility. So for example, they can go faster with mucus, and they can penetrate in host also, because of their this kind of screw kind of shape, this spiral shape, that is advantageous for them. So this is like a little bit discussion about the shape, arrangement and size of bacteria. Now I will discuss about, particularly I will focus on bacterial cell organization. There are few things which are very similar to eukaryotic cell, but there are few things which are like completely different, which are unique to bacterial cell. So here, as you can see, this is just some kind of section through bacteria, so that we can explain what is present inside the bacteria. That's why we are showing in this way. And here, as you can see at the center here, so whatever you can see here, this is nucleoid. So since you know that bacteria, they don't have organized nucleus, only the bacterial DNA, the chromosome, it is present inside the cell, and this is called nucleoid. And this one, I can say this is the bacterial chromosome. Here I should mention that almost all bacteria, they have a circular chromosome and just one in number. Some exceptions are there. For example, Vibrio cholerae, so they have 2 chromosomes. This is just some exception. Vibrio cholerae, it has 2 chromosomes. And now if you see inside the cell, we have cytoplasm, and now look into this. In cytoplasm, some kind of granular structures are there, little bit bigger in shape. This is some kind of inclusion. So they are like I would say some kind of storage body. So mostly they store different types of carbon and phosphate component. So these are called inclusion. And now apart from that, inside bacteria there are lot of ribosome, which are absolutely required. As you know that ribosomes are the major thing for translation. So in order to carry out translation, they must have ribosome inside it. And then like there are multiple layers on the surface. The innermost layer, this is called plasma membrane here. Plasma membrane, I will discuss in more detail soon. Plasma membrane. And just outside the plasma membrane, bacteria, they have cell wall. Animal cell, they don't have cell wall, right? Plant cell, they have cell wall, but bacteria, they have cell wall. And this cell wall is kind of unique. I am going to discuss in more detail soon. So this is cell wall, which is mostly composed of peptidoglycan. And now many bacteria, but not all bacteria, they have outside the cell wall some kind of capsule or slime layer. So this is the outermost layer. It can be capsule or some slime layer present just outside the bacteria. But again, all bacteria, they don't have this kind of layer. So these are some of the important stuff present in bacteria. But as you know, they don't have membrane-bound cell organelle like endoplasmic reticulum, mitochondria, whatever we have, as you already know. And apart from that, on the surface, some bacteria, they have this kind of hair-like structure. This is called pili and fimbri. They are very similar structure, hair-like structure present on the surface of bacteria. Like again, not all bacteria, they have pili or fimbri, and this helps in adhesion, and also I would say sometimes it's important for bacterial mating, so they can transfer some genetic content from one bacteria to other bacteria through this pili. And now this one is flagella. It's much bigger and robust compared to pili or fimbri. This is flagella, and flagella is involved in the bacterial movement. So flagella rotate and it's move, and that's why many bacteria, they can move from one place to another place. So these are some kind of overall organization of bacteria. And now I will discuss some of the important features of bacteria step by step. So bacterial cell membrane and uptake of nutrients. Why it is important? See, as I already mentioned, like on the surface, you have the membrane first. They are kind of separating the cytoplasm inside and the outside environment. And just after the plasma membrane, we have cell wall and sometime outer envelope. But the thing is, plasma membrane, it acts as selectable marker, like selectable permeable barrier. So why? Because cell wall and outer membrane of some bacteria, they are much more, I would say, porous in nature. So many things can pass through this. But the thing is, the plasma membrane, they act as selectable permeable barrier. So plasma membrane, as you know, that it is lipid bilayer, right? So this is very similar to eukaryotic plasma membrane. They are same. Some minor differences are there. I will mention that also. So as you can see, so here this is lipid bilayer, and this is here, this is plasma membrane, and here this is hydrophilic phosphate group, and this is I would say hydrophobic tail, So now, as a result of that, so in this plasma membrane, many things cannot cross here. Only selectively things can cross through this membrane. And also in the membrane itself, there are many protein, carrier, channel, all of those things are present to facilitate that process, right? So I'm going to discuss that, this selectable permeability across the membrane, because this is very important. Why? Because particularly bacteria, they are single cell, and whenever they are present in some solution or some natural environment, like constantly the surrounding is changing. So as a result of that, bacteria, they should efficiently control their their environment inside the cell also, like concentration of salt and concentration of nutrient, all those things, they should carefully take care of those things, right? So that's why this is very important to understand. So here, as you can see, this is much more complex organization we are showing present in plasma membrane. There are many things like glycolipid, oligosaccharide, integral membrane protein. There are many things are present on the membrane itself. And as you can see in this red color is mentioned here, haploid. So this is just I would like to mention here. In eukaryotic cell, as you know, we have sterol. For example, in our plasma membrane, we have cholesterol, right? And in case of bacteria, they don't have cholesterol. Instead of sterol, they have this haploid kind of thing. This is very similar in terms of structure, very similar to sterol, right? And this gave more flexibility to the membrane. So now, if I go to the next slide here, the bacterial cell membrane and the uptake of nutrients. So there are two major type of uptake. So I would say, so one is diffusion, and the other one is, I would say, active transport mechanism. Diffusion and active transport. As you understand, diffusion, this is not requiring energy in terms of no ATP required. Active transport, we have to spend some metabolic energy in order to carry out this kind of transportation. So diffusion particularly, here it can be passive diffusion and facilitated diffusion. So passive diffusion is only like applicable for very selected material. For example, I would say carbon dioxide, oxygen gases, they are dissolved in solution and it can go through passive diffusion inside the cell or outside the cell, right? So this is passive diffusion. But the facilitated diffusion, this is little bit complex than passive diffusion. Here, some kind of membrane protein, they are involved in this kind of diffusion, but this is both of them, like passive diffusion and facilitated diffusion. Since we are not using any energy, this should be done like on the basis of like concentration gradient, right? And in terms of concentration gradient, if some concentrated stuff present outside, it should go inside, or something which is present more inside the cell, it will go outside. So this is depending on the concentration gradient. Particularly in case of facilitated diffusion, as you can see, some carrier protein is present here. So this is a carrier protein, as you can see, this is carrier protein present on the membrane. So as a result of that, I would say this is in this membrane, this is the outside and this is inside. Similarly, here in this membrane, this is outside and this is inside. This is the same membrane, same thing is happening here. We are just showing the next step in facilitated diffusion. In the first step, as you can see, this carrier protein, they are facing outward because of some of their conformation. This is because of the conformation change in protein, it helps. So these solutes, they are entering into this carrier protein. As you can see, these are the solutes, okay? It can be some nutrients. So now, after binding solute, the conformation change, and then it can release, the carrier protein releases those solutes here inside the cell. As I already mentioned, this is the inside the cell, they are releasing solute. So as a result of that, this is one such mechanism where no energy required, but this is facilitated diffusion, not normal diffusion, right? Here some carrier protein, they are kind of facilitating this process. But although this is observed in many bacteria, I would say this is not that common, because in natural condition, if you consider, bacteria naturally present in some, some, I would say some water or in some other places, that is not that much nutrient available there, not so much nutrition rich environment. So as a result of that, it is not always easy to take nutrients from outside to inside through this facilitated diffusion method. So as if the nutrients amount is low outside, then it needs to spend some energy, and then it actually uptake those nutrients from outside to inside in that way. So here I am going to discuss that part also. That is active transport, right? So where we need to spend some energy here. So this transportation phenomena, it's very similar in eukaryotic system also, right? So here, as you can see, like in this plasma membrane, this is called, particularly this is ABC transporter. So ABC transporter means ATP binding cassette transporter. So A, B, C, ABC binding, ATP binding cassette transporter. So as you can see that on the plasma membrane, we have some specialized protein molecule present here, right? So now again I would say this is the inside, that is the cytoplasmic side, and this is periplasm outside, but it's periplasm because outside, like just after that we have cell wall also, that's why this is periplasm. So as you can see here that some solute binding proteins are available here. Solute binding protein, this is some kind of protein which will bind to solute, and then after binding to the solute, then this solute binding protein will bind to this protein, this ABC transporter, and it will send the solute inside the cell. So as a result of that, the solute will be now inside the cell. So but in order to carry out this, we need to spend ATP, as I already mentioned. This is some kind of active transport mechanism. And now this active transport can be uniport or co-transport. Uniport means here like one kind of substance going in one direction. For example, whatever I mentioned in case of ABC transporter, as you can see here, the one kind of solute is going in only inward. This is unipolar, one thing is coming. Co-transporter, here two things, two substance, like one can be ion, one can be something else, so that are moving. If both of them, they are moving in same direction, it is called symporter, and if two things are going in opposite direction, and this is called antiporter. So I just discussed this transport phenomena, but because this is very important to understand in terms of transport across the membrane. As I already mentioned, the basic mechanism is very similar to eukaryotic cell. Now just I discussed about the plasma membrane. Now I am going to discuss about the bacterial cell wall and its unique features. This is very important because of this cell wall we have lot of because of the unique features of cell wall, there are many antibiotics we use that prevent bacterial growth. So as you can see here, this is some kind of electron micrograph here. So this is some bacterial cell, and here this is more like a reconstituted image, so that this is reconstituted image, so that it will be easy for us to understand. Now as you can see, this is, I would say, cytoplasm, and now this P here, P stands for here plasma membrane. Plasma membrane. So now 1 minute. So this is PM. PM means plasma membrane. So then in this case, this is murin. P stands for here periplasmic space. Okay, and this is plasma membrane. So I am just erasing this. This is periplasmic space, the space between the cell wall and the plasma membrane. So this is periplasmic space, and this is M. M stands for here murin or peptidoglycan, same thing, murin or peptidoglycan. This is the major component of bacterial cell wall. So this is here in the reconstituted image, you can see this is the peptidoglycan or cell wall, and this is plasma membrane, and together, like as you can see, this is cell wall, you can say. Similarly, if you see another group of bacteria, so this is found in one group of bacteria. In another group of bacteria, all things are similarly labeled here. This is the plasma membrane, this is periplasmic space, which is little bit bigger, more space available here. This is the murin or peptidoglycan, and this is outer membrane. So this outer membrane is not present in this group of bacteria, right? So now, as you can see here, so particularly if I label here now, this is plasma membrane, and this is here peptidoglycan, and this is outer membrane, which is absent here. On the left side, whatever I am showing that is absent, outer membrane is not there. And together, this outer membrane and peptidoglycan here, we are saying this is cell wall, and here periplasmic space. So now, these two different types of cell wall, you can see its organization. But now, when scientists they stand with some kind of specialized technique, so these bacteria, they are called gram positive, because somehow Christian Gram, when invented this technique of bacterial staining, he found that these bacteria, they are taking dark purple color, and these bacteria are called gram positive bacteria. On the other hand, this group of bacteria, they are gram negative bacteria. This is specially happening because of their differential, different properties in their cell wall. Particularly, I would say, because of the very thick peptidoglycan layer, as you can see, this is the very thick peptidoglycan layer, and because of that, these bacteria, they behave as gram positive bacteria. On the other hand, if you see, the peptidoglycan layer is very thin in case of gram negative bacteria. So then, what is the technique here? Very briefly, I will go through it. Particularly this gram staining relies on differences of cell wall properties. So as you can see, these are the steps in staining. So as you can see, there are different types of bacteria. If you can first make some kind of smear, and then the first step is crystal violet staining. So after crystal violet staining, as you can see, all bacteria, they are violet. They are violet in color, dark violet in color, and then we add some iodine. This is called mordant. So it will fix the color. So the color retention will be better. And then in step 3, we wash those bacteria by alcohol for decolorization. So as you can see now here, some bacteria here, this is now colorless, right? This is now colorless, but these bacteria, they are still violet or purple in color. So after this step, we add some counter stain, that is safranin, which is some kind of pink color. And as you can see, those colorless bacteria, they are now pink in color, right? This is pink in color. So this bacteria, whatever pink color bacteria we are getting after this staining method, this is now, we say, gram negative bacteria, and this is gram positive. So purple here, gram positive bacteria. So as I mentioned that Christian Gram, so he invented this technique, and accordingly we named gram negative and gram positive bacteria. And this is very important step even in clinical setting. So in this way, initial characterization of bacteria can be done. Like you can just through this technique very quickly we can divide bacteria into two groups, like either gram positive or gram negative. In case of gram positive, they have like the thick peptidoglycan layer, and that is somehow preventing this decolorization step. So as a result of that, gram negative bacteria, they are getting decolorized, so they don't have the crystal violet color anymore. As a result of that, when we are adding the counter stain, in this case safranin, then they are getting pink color. So here this is some kind of real picture. As you can see, this pink color bacteria, this is gram negative bacteria. Here deep purple color, this is gram positive bacteria. So now I just mentioned about the cell wall, but I would like to mention a little bit more chemistry about the cell wall, which is very important for the for therapeutics also, for antibiotics also. So peptidoglycan, this is unique because this is not present in plant cell wall, and animal we don't have the cell wall. So as a result of that, if we can target the peptidoglycan synthesis, if we can stop peptidoglycan synthesis, then we can prevent bacterial growth also, right? So that's why it is very important. And peptidoglycan is a polymer of sugar and amino acid. Sugar here is N-acetyl muramic acid and N-acetyl glucosamine. So this is N-acetyl glucosamine, and here this is N-acetyl muramic acid. So in short, we are writing NAM and NAG. So these are the sugar. Repeating sugar is present in this peptidoglycan chain. And now this NAM or the N-acetyl muramic acid is attached to like amino acid here. So alanine followed by glutamic acid followed by diamino pimélic acid, or sometime it can be L-lysine also, depending on different types of bacteria. So now here another amino acid is D-alanine, as you can see in this figure. So this is in E. coli. So what happened? We have chain of sugar polymer and a repeating subunit like NAM, NAG, NAM, NAG. So it's repeating that way. And then some small peptide chain, mostly made up of 4 amino acid. So they are kind of hanging from the N-acetyl muramic acid, as you can see here, this way. And these amino acids are little bit different. As you can see, like when we discuss about proteins, most of those amino acids are L form amino acid, natural amino acid, which is present in protein. But here, many of the amino acid, they are D form, like D-glutamic acid, D-alanine, and that is advantageous for bacteria because they are some kind of resistance, like resistance to different peptides and so many things. So as a result of that, I would say this is bacteria like they are advantage for bacteria. Now this cross-linking is very important between this short peptide and another short peptide. As you can see, some kind of cross-linking here. This is direct cross-linking in case of E. coli. But in case of some other bacteria like Staphylococcus aureus, as you can see here, like pentaglycine, 5-glycine, they are actually helping in this cross-linking process. So as a result of that, as you can see, like particularly antibiotic penicillin or its derivative, they stop this cross-linking process, and therefore peptidoglycan cannot be synthesized, and that's why it acts as antibiotics. So whatever I discuss about this peptidoglycan structure, little bit you will get kind of more three-dimensional understanding if I draw this in this way. So I would say NAM, NAG, NAM. So it's going like this. Similarly, NAM, NAG. So this way, these are the sugar polymer. Now from NAM, we have this L-alanine and then D-glutamic acid and then L-lysine and D-alanine. Similarly, from here also this N-acetylmuramic acid also you have the similarly this peptide L-alanine, D-glutamic acid, L-lysine and D-alanine. Now what happened? This D-alanine is attached to this L-lysine by pentaglycine in case of Staphylococcus. This is the cross-linking. Similarly, again like peptide will be hanging from this NAM and from here also hanging like this, and again this cross-linking. So this way it will make up kind of strong mesh kind of thing, and this layer is present in much more quantity in case of gram-positive bacteria compared to the gram-negative bacteria. That's all. Now, some special features I am going to discuss about bacteria. So, as we know about cytoskeleton is present in eukaryotic cell, for example microtubule, actin and intermediate filament, but current research also revealed that in bacteria they also have some kind of cytoskeletal material, as you can see this one FTSZ. So, this is very similar to, I would say, homologue of tubulin. So, this is similar to eukaryotic tubulin. So, and it helps in cell division and widely observed in many bacteria and archaea. So, for example, we are showing here in bacillus. So, bacillus are a rod-shaped bacteria, it is in bacillus. And another cytoskeleton you can see here MREB or MBL. This is again some kind of protein maintaining the cell shape and localizes protein inside the cell. This is also present, and this is, I would say, actin homologue. So, this is present in bacteria. So, this is very interesting. As you can see here, this is GFP level here, GFP level FTSZ, and this is GFP leveled MBL protein present in bacillus. And another interesting, like some special features in bacteria is some bacteria they have some kind of small structure primarily made up of magnetite, like as you can see here, and help in navigation through Earth's magnetic field. As you know that, like for example, many different animal, I would say particularly some bird and dolphin and some fish like tuna. So, those animal they have some kind of magnetic setup inbuilt in their head, and accordingly they align their magnetic pole, and accordingly they can move, they can migrate from one side to another side, right. Similarly, some bacteria they have also this kind of magnetosome, and that help them to align particularly and move through the Earth's magnetic field. It's very interesting. So, this is, see, bacteria, they are so simple thing, just unicellular organism, but still they have this kind of complexities, like, and this MAMK, this protein, this is again another cytoskeletal protein which is responsible for the orientation of this magnetic particle inside this bacteria. And this is, we are just showing one magnetotactic bacterium. And now few things here, like bacterial some special features, for example capsule and slime layer. So, capsule and slime layer, this is, I would say, present in some bacteria. For example, during Griffith's experiment I discussed about Streptococcus pneumoniae. So, they have some kind of layer, polysaccharide layer, right. So, this is mostly made up of polysaccharide in Streptococcus pneumoniae. So, it helps to help in evading immune system. So, immune cell cannot quickly phagocytose those bacteria. So this is again some kind of advantage for bacteria, and sometimes it also helps in attachment. And also bacteria, they have pili or fimbri, as I already mentioned. So here they have small hair-like structure, and they again help in adhesion, sometime in mating, like this is called sex plus or F plus. So this is help in mating. And now flagella, which are much bigger and robust compared to pili or fimbri. As you can see, some bacteria, they might have just one flagella, or they might have multiple flagella, and they help in movement of bacteria. And also inside the flagella, so they have some kind of motor, and which rotate very, very quickly. So this is kind of astonishing number. If you see here, 200 to 1200 revolution per second. So this is, again I am saying, revolution per second, so fast they can move. And based on their movement, based on their direction of the movement, different types of motion can be observed in bacteria. And here, just we don't need to go through the details of the structure, but here I am trying to show that how complex system can be integrated even in a small cell like bacteria. So this is the organization of flagella. So as you can see, this is the motor, right? Motor present in the base of flagella, and it requires some energy to help this motor to rotate, and particularly I would say this proton, like this proton movement, that helps the movement of this motor also, and it helps bacterial movement. This is very important. And although flagella is not present in all bacteria, but many bacteria, they have flagella. And at the end, this is another one, endospore. So endospore is present in some bacteria. It is very medically important and challenging for medical professional also, because this endospore present inside bacteria, it is very much resistant to, like I would say, to heat, to different types of radiation, including gamma radiation. So and many chemical substances, like generally we use to like most of the chemical you cannot really kill by this, like disinfectant you cannot use to kill endospore, and they are very resistant. And this is present in some bacteria like Clostridium botulinum, Clostridium tetani, which causes tetanus, and the Clostridium botulinum, here we have like botulinum toxin, which is very, very toxic, like a potent toxin. So as a result of that, this is very challenging to take care of this endospore problem, right? So that's all. Here like something mentioned here, horizontal gene transfer mechanism in bacteria and its implication. So it will take a little bit more time. I will discuss in the in some other class. So that's all. And like you can refer any of these textbook. Mostly I cover from this textbook, and some informations are also present in this book as well. Thank you very much.